Hello, dear guys. So here's the third lecture in pathology review. So it's also just introduction to the pathology, and we continue our set of lectures here. It's Olympic University. So my name is Knyazov Igor, as written here. So let's move forward. We stopped on the topic of cellular metabolism and mitochondrial function. So cellular metabolism is made by almost all the actions of cellular metabolism is provided with the help of mitochondria. So let's talk about mitochondria. So in more details, uh, you didn't discuss it in histology or somewhere else. So, mitochondria evolved from ancestral prokaryotes that were engulfed by primitive eukaryotes about 1.5 billion years ago. So, it's prokaryote. Their origin explains why mitochondria contains their own DNA genome. It's circularized, so like prokaryotes do. About 1% of the total cellular DNA is present inside the mitochondria. It encodes approximately 1% of the total cellular proteins and approximately 20% of the proteins involved in oxidative phosphorylation. So oxidative phosphorylation, I remind you, it's your beloved Krebs cycle, and which resulting in formation of NADH, FADH, and also formation of ATP in the end. Although their genomes are small, mitochondria can nevertheless perform all the steps of DNA replication, transcription, and translation. So mitochondrial machinery is similar to present-day bacteria. For example, mitochondria initiate protein synthesis with N-formylmethionine and are sensitive to antibacterial antibiotics. Mitochondria are dynamic, constantly undergoing fission and fusion with other mitochondria. Okay, in this way. Anyway, mitochondria are dynamic, constantly undergoing fission and fusion. In this way, mitochondria can undergo regular renewal to stave off degenerative changes that might occur because of genetic disorders or oxygen-free radical damage. Mitochondria turn over rapidly with estimated half-lives ranging from 1 to 10 days depending on the tissue, nutritional status, metabolic demands, and intercurrent injury. Because of the ovum contributes the vast majority of cytoplasmic organelles to the fertilized zygote, mitochondrial DNA is virtually entirely maternally inherited. However, because the protein constituents of mitochondria derive from both nuclear and mitochondrial genetic transcription, mitochondrial disorders may be X-linked, autosomal, or maternally inherited. So, conclusion. So, mitochondria are delivered to us from the maternal egg. So ovum, which fused with sperm that time during the fertilization process possess their own mitochondria, and mitochondria from sperm don't enter inside the zygote. That's a point that mitochondria might inherit. It's 1% of DNA which is carried by mitochondria is derived from mother, and that's why we can conclude this so we used to say that half of genetics came from mother, half from father, yeah, but actually a little more, approximately. So now we can conclude that 51% is given from mother, and 49 is given from father, because 1% of DNA is given by mitochondria. You should remember this. Actually, now we have a lot of researches, and so also we can that DNA actually changes after every replication a little bit, yeah. So not it is not so tremendous changes in DNA consequence during every cell division, every mitochondria fission, but we can follow this change to conclude so where the uh, uh, so we can find relatives of this mitochondria in ancient people and pre-ancient period, and by this we followed the changes of mitochondria, and we found that we have one common mitochondria type with chimpanzees. Then before the chimp, so in other apes, yeah, and that's by with help of this mitochondria we can conclude that we are derived from animals and from apes. So if still someone of you is thinking that humans are came from from I don't know, not from monkeys, but they are we are so because evidence we have from mitochondria DNA changes shows us that we are derived from animal world. So without any questions, okay, let's continue. Let's talk about function of mitochondria. Mitochondria provides the enzymatic machinery for oxidative phosphorylation, and thus the relatively efficient generation of energy from glucose and fatty acid substrates. They also have central roles in anabolic metabolism and the regulation programmed cell death, so-called apoptosis. So, especially next set of lecture will be about cell death, the cell changes during cell changes due to cell stress, and mitochondria, of course, react on it as one of the first cell. So let's talk about the structure of mitochondria. So we see the outer membrane and inner membrane. So outer membrane is pretty straight, yeah. So it's a smoother appearance. Inside the outer cell membrane we have proteins like pumps and channels. Which help them to be a little insulated from the cytoplasm, and possess their own cytoplasm here in between these two membranes. So it is known as intermembrane space. And actually, in this intermembrane space, actually ATP is generated, and then when ATP is generated here, it's transmitted into the cytoplasm, to the sites of action, and gives the energy to our cellular changes. So, for example, the first example which comes to my mind is sodium potassium pump. It requires ATP energy, and ATP, which is used there in cell membrane, sodium potassium pump, are produced in mitochondria. That makes mitochondria one of the main organelle which facilitates the cell surveillance. So okay, let's and we have inner membrane. Inner membrane is so invaginated. Yeah, space of inner membrane is much larger than outer membrane. So here it's shown like small projections inside the matrix, yeah, mitochondrial matrix, also known as core matrix. Yeah, and that's necessary for what? Because this inner cell membrane possesses a lot of enzymes of this Krebs cycle, and inside the matrix we also have enzymes which facilitate the Krebs cycle. So this approximate structure we will continue discussing a little later. Main idea is that mitochondria produce H plus ions, so protons, yeah, and it also possesses a lot of group of family of proteins which are known cytochrome proteins, and especially cytochrome C. And when we have, so we'll discuss it in next lectures. So in some next lecture about cell death, and both of these channels are used to optimize the cell death. And we have necrosis process, so when some acute injury happened to the cell, so when H plus ions enter into the cytoplasm, and you understand H plus is acidic, so it provides the acidic milieu, and increasing of acidic milieu change the metabolism of the cell, which finally causing the uncontrolled cell death, like necrosis. And mechanism of this also gonna be discussed later. And another pathway of cell death, which is maintained by mitochondria, is the delivery of cytochrome C from the matrix of the mitochondria. So it releases into the cytoplasm, finds specialized proteins. So it's also multi-step changes, but main idea is that cytochrome C binds to apoptosome, and apoptosome is formed, and due to this, apoptosome activates the mechanism of another type of cell death. It's known as apoptosis. So it activates the caspase cascade, which also be discussed in one of the next lectures. But you see, mitochondrion is involved in both types of cell death, and one of the key processes. So in different types of cell deaths are located in mitochondria. Okay, let's talk about functions. Main function of mitochondria is energy generation. We used to name the mitochondria is the powerhouse of the cell. Yeah, and that it. Each mitochondria has two separate and specialized membranes. The inner membrane contains enzymes of the respiratory chain folded into crista. This includes the core matrix space that harbors the bulk of certain metabolic enzymes, such as enzymes of the citric acid cycle. Everything I explained to you right now. Outside the inner membrane is the intermembrane space site of ATP synthesis, which is in turn enclosed by the outer membrane. The latter is studded with porine proteins that form aqueous channels permeable to small molecules. Larger molecules and even some smaller polar species require specific transporters. Everything like in usual cell membrane. The major source of the energy needed to run all basic cellular functions derives from oxidative metabolism. Mitochondria oxidize substrates to CO2 and in the process transfer high energy electrons from the original molecule, like in glucose, to molecular oxygen to water. Oxidation of various metabolites drives hydrogen ion, like proton, pumps that transfer H plus ions from the core matrix into the intermembrane space. This process is H plus ions flow back down the electrochemical gradient. The energy released is used to synthesize ATP. So nothing new for you here. Let's move forward. It should be noted that the electron transport chain need not necessarily be coupled to ATP generation. So here it's not shown. Sorry, here it's not shown, but you know that when you have fever, so what actually happened? So these H plus ions we have two pathways to generate energy: to generate heat energy and to generate the ATP energy. And when it goes through the ubiquinol system of chain, so it produces the ATP. But if it skips the ubiquinol system, so H pluses are they directly bind to oxygen and producing the water, and that's how we use oxygen actually in this metabolism. And when they directly binds, so you remember from chemistry that when reaction goes, exothermic reaction goes, so heat is released. And we have a choice. Yeah, mitochondria has a choice to produce ATP energy or heat energy. So I hope it's understandable. And some hormones like thyroid gland hormones, so which is located here, yeah, it produces the tyrosine and triiodtyronin. This tyrosine, triiodtyronin, thyroxine and triiodtyronin, sorry, it actually skips the ubiquinol system and causes the heat. Keep it in mind, it's a site of action of some hormones. Mitochondria is a site of especially thyroid hormone activity. Okay. Natural byproduct of substrate oxidation and electron transfer, mitochondria also are an important source of reactive oxygen species. So, oxygen, which is involved in metabolism of mitochondria, sometimes producing the reactive oxygen species, which are later we'll name it as ROC, ROS, and I will name it in this way. So please remember this abbreviation, reactive oxygen species. And they can produce oxygen-free radicals, hydrogen peroxide, and even nitrogen oxide. So we'll talk about today a little later. And it causes the oxidative stress. So because it's radical oxygen species, they are able to destroy cell membranes. All types of cell membranes, and one of them is lysosome membrane might be destroyed. And you remember that lysosome possesses hydrolytic enzymes in it, and it leaks when it leaks out. It's going to destroy not only cell membranes but everything which is in cell. So it is causing of necrosis. Also, we are discussing in proper topic. Intermediate metabolism. That's what I talked about right now. So please, if you didn't understand me there, so make a pause and read it. Cell death. Mitochondria are like the proverbial Dr. Jekyll and Mr. Hyde. On the one hand, they are factories of energy production in the form of ATP that allow the cell to survive. On the other hand, they participate in driving cell death when the cells are exposed to noxious stimuli that the cells cannot adapt to. The role of mitochondria in the two principal forms of cell death, necrosis and apoptosis, will be discussed in next lectures. In addition to providing ATP and metabolites that enable to bulk of cellular activity, mitochondria also regulate the balance of cell survival and death. Cellular activation. Cell communication is critical in multicellular, so it's in the next chapter of this lecture. Cell communication is critical in multicellular organisms. At the most basic level, extracellular signals determine whether a cell lives or dies, whether it remains questioned, or whether it is stimulated to perform a specific function. Intercellular signaling is important in the development of embryo, in maintaining tissue organization, and in ensuring that tissues respond in an adaptive and effective fashion to various threats such as local tissue trauma or systemic infection. Loss of cellular communication, the social controls that maintain normal relationships of cell can variously lead to unregulated growth, which is known as cancer, or an ineffective response to an extrinsic stress, like in shock. Cell signaling, as an individual cell is constantly exposed to a remarkable cacophony of signals, which must be interpreted and integrated into responses that benefit the organism as a whole. Some signals may induce a given cell type to differentiate, others may stimulate proliferation, and yet others may direct the cell to perform a specialized function. That's signaling. That's a function of receptors. Multiple signals received in combination may trigger yet another totally unique response. Many cells require certain inputs just to continue living. In the absence of appropriate exogenous signals, they die by apoptosis. So, some example of it is, for example, in prostate. Yeah, so I'm a urologist. I give these examples. So a lot of prostate cells, they are charged to intake of testosterone molecule, so this hormone, and when testosterone level is high, so prostate is living their life and proliferate, and everything keeps in normal. But when level of testosterone is skipped, so drop down on the basic level, so it causes the shrinking of prostate and replacement of prostate tissue into the connective tissue. So, and we use this feature in treatment of prostate cancer. So we just give the to patient blockers of testosterone synthesis enzymes, and testosterone level skipped, and cancer cells of prostate is also dropped. Also, like option, we can make the castration. So we just remove the testicles from patients' scrotum, and it also gives a good result in treatment of prostate cancer. Yeah, it's not the only way to treat. You understand that testosterone is necessary for young men, and so usually, sometimes patients try to avoid this kind of treatment. That's why we have another types of treatment. But this is very efficient way to treat this type of disorder. Okay, pathogens and damage to neighboring cells. Many cells have an innate capacity to sense and respond to damaged cells. They're known as danger signals, as well as foreign invaders such as microbes. The receptors that generate these danger signals are discussed in lectures of inflammation and repair and in immune system diseases. So cell-cell contacts mediated through adhesion molecules or gap junctions. As mentioned previously, gap junction signaling is accomplished between adjacent cells via the hydrophobic connexons that permits the movement of small ions like calcium, various metabolites, and potential second messengers such as CAMP is the cyclic adenosine monophosphate. Cell extracellular matrix contact. They are mediated through integrins, which will be discussed in the inflammation chapter, in the context of leukocyte attachment to other cells during inflammation. So also keep it in mind. Secreted molecules. The most important secreted molecules include growth factors, discussed later, cytokines, a term received for mediators of inflammation and immune responses, and hormones, which are secreted by endocrine organs and act on different cell types. Signal pathway can be classified into different types based on their special relationships between the sending and receiving cells. We have paracrine signaling, autocrine signaling, synaptic signaling, and endocrine signaling. Paracrine signaling. So when one cell producing some hormone-like messengers to the neighboring cells, and they control this one controlling cells, control cells, and behind. So they are might be normal excretion might be happened in inflammation reaction, for example. And all of you remember from physiology that we have some mast cells, yeah, that derive from blood, cells that derive from blood, and they producing the histamine, bradykinin, and serotonin, and they control the cells behind them. So they don't produce it into the blood; they produce it into the neighboring cells. Then we have a specific type of this specific type of secretion is autocrine signaling. Autocrine signaling is when a cell itself produces some molecules to stimulate the cell itself, and we have it is used to amplify activity of the cell. For example, cell is active and it producing more signals to activate the cells more time. Or it also used to in dampen. It's like known as negative feedback response. So when cell is overproducing something and already extracellular matrix is fulfilled with the secretive molecules, so it gives a negative feedback to give the signal to stop producing all these molecules, just as example. Then we have synaptic signaling. Activated neurons secrete neurotransmitters at specialized cell junctions onto target cells. This is well discussed in nervous system. So when a neuron is given to tissues and synapses attached to them, for example, in muscles. Yeah. So when motor neurons are attached to muscle cell, skeletal muscle cell, it directly gives a stimulus to this muscle cell to contraction. It's a point of acetylcholine receptors, acetylcholine receptors on the muscle, which are activated by neurons of the synapse of the neurons of the by motor neurons of the spinal cord, for example, if we're talking about the body. And they have target cells and these receptors on them. And we have endocrine signaling. So endocrine signaling is the most convenient thing to discuss. So most of you already heard about it. So it's endocrine system is working on this. That we have some cells that have receptors to some hormone, and we have specialized organs which produce these endocrine signals. They produce hormones, and through the bloodstream, we so on the large distance, we can control different organs. So best example is our pituitary gland, which is located in brain, but it activates sometimes all the cells of human body. For example, in the growth hormone, somatotropin hormone, and somatropic hormone, and also they can act on different or some another endocrine organs. For example, gonadotropic hormone, so which is released from pituitary gland, so activates uh, our sexual organs activity, and they can okay. It was discussed in physiology very well. So, but still keep it in mind. Regardless of the nature of an extracellular stimulus, paracrine, synaptic, or endocrine, the signal it conveys is transmitted to the cell via a specific receptor protein. Signaling molecules, which are like hormones, they're known as ligands, bind their respective receptors and initiate a cascade of intracellular events culminating in the desired cellular response. Sometimes not desired, but it conveys a response. Ligands usually have high affinities for receptors, and at physiological concentrations bind receptors with exquisite specificity. Um, Receptors may be present on the cell surface or located within the cell. We have intracellular receptors. So how intracellular receptors act? They include transcription factors that are activated by lipid-soluble ligands that easily transmit plasma membranes. So you remember structure of plasma membrane that in 50% it composed of lipids, in 50% it composed of proteins. And through this lipid portion, so other lipid-like compounds can easily penetrate down into the cell. So this example, it includes vitamin D and steroid hormones. They activate nuclear hormone receptors. Um, in other settings, a small and or non-polar signaling ligand can diffuse into adjacent cells. So example is nitric oxide. So endothelial cells regulate intravascular pressure by them. Nitrogen oxide is generated by an activated endothelial cell, blood vessel cell, yeah, so which in direct contact with the blood, and then diffuses into adjacent vascular smooth muscle cells. And the smooth muscle cells responding by dilation of blood vessel. And a good example here is dilation of our coronary arteries. Coronary arteries in cases of so we can activate them by ourselves, so with the injection of nitrogen oxide donors. So for example, you know that compound, it is trinitroglycerin. So we prescribe this for patients with the heart attacks to in desire to help them to equip the heart with a larger amount of blood. So okay, that's is the point of later discussion. Cell surface receptors. Cell surface receptors are most known. They're transmembrane proteins of the cell membrane with extracellular domains that bind activating ligands. Depending on the receptor, ligand binding may do these actions. They can open ion channels. For example, at synapse between electrically excitable cells. So that's acetylcholine, as I said before. So it opens the sodium channels and activates the active potential afterward. Secondary, activate an associated GTP binding regulatory protein, known as G protein. It is discussed later. Activate an endogenous or associated enzyme, often tyrosine kinase. That's a very common type of response to hormone activation. And number four is a specific type for which is used in embryonic development. Trigger a proteolytic event or a change in protein binding or stability that activates a latent transcription factors. So this number four type of receptor actually change the fate of the cell. It changes the genetic factors. So it activates some parts of genes which used to be silent in the cell, and that's why the cell gets some extra features. So especially in process of differentiation of cells, it is extremely important thing. As I noticed you in some previous lectures, that stem cells we have, which are derived from the um, which came to us in embryonic period. They can be activated by this number four type of receptors that are that activate some genetic expression of the cell and make them what they are. Once again, so you know that we have every cell of human body is possessing absolutely similar amount of DNA, similar amount of genes, but different expression of these genes explains the difference in their functioning in a body, which makes a muscle cell as a muscle cell, which makes skin cell, epidermis, I mean, and brain as a brain cell, neuron as a neuron. And the dysregulation of this number four type of signaling causing the disorders like cancer, so which we also discuss in chapter number 4 in lectures which will be discussed in later topics, especially when we will discuss about the tumors. There are examples. Yeah, that ligand is usually a small molecular compound which binds to receptor, and it causes the activation of non-receptor tyrosine kinase. We have some receptors which are tyrosine kinase itself and ligand binding to them, so activate them. So actually functionally they are like similar, but biochemically, by molecular biology, they are quite different. Now we have some receptors which activate the cAMP pathway. I said you before, cAMP is cyclic adenine monophosphate. So ATP is converted to cAMP, and it also causes a lot of changes inside the cytoplasm. So also before we discuss about the nuclear hormone, which passing through the lipid bilayer and enters inside the cytoplasm, where it meets the hormone receptor inside the cytoplasm or on the surface of a nucleus membrane, and it activates the transcription factors or deactivates the transcription factors, but somehow it changes the genetic expression. And here number 4 is a Notch ligand, which so usually is located on other cell, and it binds to this cell, which causes the cleavage of Notch receptor and cleaved Notch part. So it is activator, it's a transcription factor itself, and it activates the transcription of Notch target gene. So one notch is charged to activate some specific part of the DNA. Then we have freeze type of receptors. So, okay, that we'll discuss a little later. So this is activation of growth factor. So about growth factors, we have different parts in this lecture. Yeah, we will discuss it in next lecture actually in more details. Signal transduction pathways. Binding of ligand to a cell surface receptor mediates signaling by inducing clustering of the receptor. It's known as receptor cross-linking or other conformational changes. The common theme is that all of these perturbations cause a change in the physical state of the intracellular domain of the receptor, which then triggers additional biochemical events that lead to signal transduction. Cellular receptors are grouped into several types based on the signaling mechanisms they use and the intracellular biochemical pathways they activate. So once again, receptors associated with kinase activity, I show you. Receptor tyrosine kinase. So functionally they are like similar, but as I said before, here we have protein apart which activates the kinase activity, non-receptor kinases. Non-receptor tyrosine kinases are joined with activity of receptors associated with kinase activity. And then we have receptor which is itself tyrosine kinase. Also, we have G protein coupled receptors. G protein is used in the formation of GMP. So, cyclic GMP is guanylene monophosphate receptors, which also help in some events of activation of cell. We have nuclear receptors, I shown you before, so which activation of which change the genetic expression of the cell. And we have notch receptors and Frizzle family receptors. So I approximately explained them in previous slide. So receptors associated with kinase activity. Phosphorylate specific tyrosine residues, whereas serine threonine kinases add phosphates to distinct serine or threonine residues, and lipid kinases phosphorylate lipid substrates. For every phosphorylation event, there is a potential counter-regulatory phosphatase, an enzyme that can remove the phosphate residue and thus modulate signaling. Usually, phosphatases play an inhibitory role in signal transduction. That's it. Receptor tyrosine kinase are integral membrane proteins. So examples here are receptors of insulin, epidermal growth factor, and platelet-derived growth factor. Ligand-induced cross-linking activates intrinsic tyrosine kinase domains located in their cytoplasmic tails, as noticed before. And non-receptor tyrosine kinase is some kinds of receptors they have no intrinsic catalytic activity, and we need the non-receptor tyrosine kinase to activize it. Okay, let's keep it. There are a lot of molecular biology stuff here. If interesting, you can read it. G protein coupled receptors, as I said, they produce GTPs and form the GDP, or it itself it downstream signal typically involves generation of CAMP, cyclic adenosine monophosphate, and inositol 1,4,5-3-phosphate (IP3) pathway, so which is activized by G protein coupled receptors. Okay. And we have nuclear receptors, I explained you before. And we have Notch family and VNT protein ligands, they also known frizzled, that can stabilize beta-catenin, allowing it to translocate to the nucleus and form a transcription complex. So you see these receptors, other types of receptors are responsible for transcription complexes, activation, and expression of genes. So modular signaling proteins, hubs, and nodes. So which functions are made by this protein, by these receptors? Their enzyme activation or inactivation. Anyway, so it is controlled by hormones. Nuclear or cytoplasmic localization of transcription factors. So we discussed it. Transcription factor activation. Also, a transcription factor, so once again, control of gene expression. We can activize or deactivize. Single. Actin polymerization or depolymerization. So actin is a part of cytoskeleton, and we control the shape of the cell with the help of the receptors. And we can, for example, in smooth muscles, yeah, when as I noticed before, so nitrogen oxide, so they can change the actin polymerization complexes, and which leads to contraction or but in cases of nitrogen oxide relaxation of smooth muscles, so with the help of depolymerization. Protein degradation or stabilization is controlled by this, and activation of feedback inhibitory or stimulatory loops. So also I explained on the example of um, autocrine secretion. Adapter proteins. The adapter proteins play a key role in organizing intracellular signaling pathways. These proteins function as molecular connectors that physically link different enzymes and promote the assembly of complexes. Adapters can be integral membrane proteins or cytosolic proteins. Typical adapter may contain a few specific domains. So, in example, it's SH2 or SH3, and so these complexes are just joined together to form some other molecule which do some activity inside the cell, so that mediate protein-protein interaction. By influencing the proteins that are recruited to signaling complexes, adapters can determine downstream signaling events. So adapter proteins, they are not receptors itself, but they are acting how it is ordered by receptors. So here the adapter protein, so in explanation of computer science, I'm not good in computer network science, but here is given this analogy. By analogy with computer networks, the protein-protein complexes can be considered nodes, and the biochemical events feeding into or emanating from these nodes can be thought of as hubs. Signal transduction can therefore be visualized as a kind of networking phenomenon. Understanding this high order complexity is a province of systems biology involving a marriage of biology and computation. Actually, we have a science which is known as so biological informatics. Biological informatics, which teach this, so which learn the relations between, sorry, a relation between information which is given to the cell and transduction of information from one cell to another, and transmission it inside the cell. Okay, and finally, let's talk about the transcription factors. There are transcription factors. They may drive the expression of a relatively limited set of genes, or may have much more widespread effects on gene expression. Among the transcription factors that regulate the expression of genes that are needed for growth are MYC and GUN, whereas a transcription factors that trigger the expression of genes that lead to growth arrest is P53 gene. So P53, so is used in checkpoints of the cell cycle. So there. Okay, it says transcription factors have a modular design, often containing domains that bind DNA and others that interact with other proteins, such as components of the RNA polymerase complex required for transcription. DNA binding domains permit specific binding to short DNA sequences, so whereas some transcription factors binding sites are found in promoters close to the site where the transcription starts, it is now appreciated that most transcription factors bind widely throughout genomes, including to long-range regulatory elements such as enhancers. Enhancers function by looping back to gene promoters and therefore are specially located close to the genes that they regulate, even though the terms of genomic sequence that they may appear to be far away. These insights highlight the importance of chromatin organization regulating gene expression, both normal and pathologic. And for a transcription factor to induce transcription, it must also possess protein Protein-protein interaction domains that directly or indirectly recruit histone-modifying enzymes, chromatin remodeling complexes, and most importantly, RNA polymerase as a large multi-protein enzymatic complex that is responsible for RNA synthesis. Second. Okay, thank you for attention. This lecture is kind of short, but need to be understandable. Thank you.